Okay, Biology B students, here we go. So this module has to do with evolution and how life kind of evolves on the planet. So here we go, the origins of life. If you look at the right here, back in the day, before Darwin and all that, there was a time where they thought things could, or living creatures can come from non-living things. So for example, if you look at the recipe for mice, you get a bowl, you get a shed, place a dead bull on branches and herbs inside the shed, wait for the summer, and all of a sudden you wind up getting all kinds of stuff. So anyway, there's lots of different things for bees. There's how do we get mice? You get some things and throw them into a yard with some towels and some food, and all of a sudden you got mice, right? But we know better at this point in time. You just simply can't get living things to come from nothing. So that was the idea of spontaneous generation, or being able to produce life from non-living things, known as abiogenesis. Is the idea that living things can be produced from non-living things. From the time of the ancient Romans through the Middle Ages and until the late 19th century, it was generally accepted that some life forms arose spontaneously from non-living matter. So the origins of life. Where did life come from? Where did life forms such as mice, maggots, and microbes, where do they come from? Could they come from nothing? Right? Again, there's all kinds of little recipes that you could see to try to produce creatures back in the day. Theory one, spontaneous production of mice required placing sweaty underwear and husks of wheat in an open mouthed jar. Wait 21 days. During that time, the sweat from the underwear would penetrate the husks of the wheat, changing them into mice. Theory two, maggots arise spontaneously in rotting meat. In theory three, microorganisms arise spontaneously in broth. We know nowadays, if you have meat outside, flies will land on them and lay their eggs, and all of a sudden maggots come. We know that in the broth, there are potential to have bacteria, and bacteria will just grow because it has a food source. Different scientists tested these theories over the years. Francesco Redi, Italian doctor, Lazzaro Spallanzani, Italian scientist, Louis Pasteur, a French scientist. You can watch some videos if you are looking through the presentation on your own. So here's Redi's experiment. So first he put flies or meat into a jar, and he left the jar open. And then at some point in time, flies would land on it and they would produce the maggots. Then he took the same thing, he put the meat in the jar, but he put gauze on top of it. No maggots appeared in the gauze covered jar. So there was no maggots that were going to be found in here. Okay, so the flies couldn't land and lay their eggs on the meat. Okay, and then finally he sealed the jar and no maggots appeared in the jar at all. So jar one open to the air, jar two covered with netting, jar three sealed off completely. The conclusion, hypothesis was supported by the data and therefore accepted because there were no maggots in or around the jar. It was concluded the maggots came from the fly eggs. Spallanzani's experiment. What causes microbes to form in the decaying broth? His hypothesis, microbes come from the air. Boiling will kill the microbes. So Spallanzani put the broth into four flasks. He left one open. He sealed the second one. The third one was boiled and left open. And the fourth one was boiled and then sealed. What would you think would happen to all of those? Would any microorganisms be found in this one? Would any microorganisms be found in the sealed jar? What about 3 and 4? The hypothesis was accepted because the combination of boiling the broth, so you kill the bacteria in the beginning, and then sealing it, it's one of the ways we can things today. It's how we can keep Campbell's soup from getting bacteria inside of it. Okay, so that hypothesis was accepted as well. And then finally, the one that was the most important one, because the critics of Spallanzani and Reddy were like, well, you got to leave the factor from the air. That's what's important in the air. It has to be able to touch the meat or the broth. And so to get around it, Louis Pasteur came up with an ingenious method, and it was this curved flask. And so now this right here would allow air to get in here, but the microbes would get stuck here and never be able to come and enter into the broth. So he was able to quiet down the critics saying you can't have it sealed for the spontaneous generation theorists, and that he did that. He didn't seal it, and he allowed air into it, and you'll see. So where do microbes come from again? His hypothesis: microbes come from cells of organisms on the dust particles in the air and not from the air itself. And so here's what he did. He had a control group, one flask of broth open to the air, an experimental group with the S-shaped flask that prevents dust from entering the flask. The air, the vital factor, which the spontaneous generations theorists said you needed to have in order to create life from non-life, was important to the process. Pasteur tried to solve this with the curved flask. So once again, this is where everything got caught. And then he tipped the broth after nothing grew in it for months and allowed some of it to come into this area, pick up the microbes, and then they wound up growing again. So if the neck is later broken, the outside air can carry microbes into it. So this was a very important one, and it kind of did put the spontaneous generation theorists to bed. So Pasteur's experiments silenced the spontaneous generation proponents for good. The hypothesis was supported because the broth did not turn cloudy and the dust was observed trapped in the neck of the S flask. It was concluded the microbes entered being attached to the dust particles. Life has to come from life. Bio, life, Genesis to begin. So life comes from other living things, like we learned about with the cell theory, right? Cells come from other cells, pre-existing cells. So how did life get here? Well, for a really long time on this planet, 700 million years, Earth was not conducive to life. Very hot, no oxygen, molten state, and so forth. Over time, the materials making up the Earth separated into the Earth's layers, crust, mantle, and core. Gases released from the Earth's interior formed an atmosphere. Now we can capture and collect and keep certain gases inside the Earth's atmosphere. So then we had these experiments. These guys were able to come up with that were very important to trying to figure out how did life start on the planet. So the theory of how life may have developed on the early Earth, based on assumptions that there was very little oxygen. We didn't have any plants to produce oxygen. The atmosphere was mainly formed from volcanic vapors, methane, which is just carbon and hydrogen, ammonia, hydrogen, water vapor. Felt it would be possible for inorganic molecules to be converted into organic forms using energy from the sun, and in that time, lightning. At the time, no effective way to test this. Miller and Urey tested Oper and Haldane hypothesis. They simulated the atmosphere containing gases, the ones that Oper and Haldane had thought were present. He exposed the gases to electric shocks, simulating electricity or lightning, and produced small organic compounds, mainly amino acids, which are, again, part of building up proteins and building blocks of life. So you can look at the experiment right here if you want. You can take a look at the whole thing right here. So the meteorite hypothesis. Analysis of meteorites indicate that organic molecules can be found in space. This suggests the following possibilities. Amino acids may have been present when Earth formed. Organic molecules may have arrived on Earth through some different types of meteorites and asteroid impacts with the planet. The formation of the first cells. Once organic molecules, compounds are formed, how did they get packaged into cells? So iron sulfide hypothesis, all right, this iron sulfide bubbles hypothesis and the lipid membrane hypothesis are two different theories. The iron sulfide bubbles, iron sulfide rising from this deep sea vents combine with cool ocean water to form chimney-like structures with many compartments. Biological molecules may have combined inside these compartments, which acted as membranes. With the right combination of ingredients, the first organic cell membranes may have been formed. The lipid membrane hypothesis. Lipids spontaneously form membrane-enclosed spheres called liposomes. This happens naturally. Liposomes could act as membranes around a variety of organic molecules, separating them from the environment. The first genetic material, it has been hypothesized that RNA was the genetic material for the earliest life forms, not necessarily DNA. So Cech and Altman in 1980 discovered the RNA can catalyze reactions, make reactions happen, and copy itself. The first eukaryotes, the true cells, the ones with the nucleus and some in the organelles, fossil evidence indicates the first living things were prokaryotes, bacteria. Okay, first appeared about 3.5 billion years ago. Eukaryote cells with the nucleus and organelles don't appear until about 1.5 billion years ago. The endosymbiosis, which we talked about a little bit, Lynn Margulis, idea that mitochondria and chloroplasts used to be simple prokaryotic cells that were engulfed by larger prokaryotes to help them so that they would do jobs for the cell and the cell would provide them food or protection or what have you. So you can watch a little video on that. Again, you can see this is a cyanobacteria. This one produces some energy, right? Maybe it was engulfed by another cell, a eukaryotic cell. Now the energy that is used can help basically be like a battery to the cell, and the cell can provide it food and it can provide it some protection. So what evidence supports this theory? Both mitochondria and chloroplasts have their own DNA, have their own ribosomes, can copy themselves, are about the same size as prokaryotes, have DNA in the shape of a circle like the bacterial and prokaryotic DNA. Why would they, no other organelles necessarily have that, so why would they have it unless they were living on their own? Okay, that ends this lesson. Stay tuned for 4.2.